Hello, this is Daniel Warila, and this is my biochemistry presentation on the article Live Cell Lipid Biochemistry Reveals the Role of Diacylglycerol Side Chain Composition for Cellular Lipid Dynamics and Protein Affinities. Okay, so background, why are lipids important? Lipids are important because they can function as enzyme cofactors, allosteric modulators, and receptor ligands in the membrane. And they're able to control cellular excitability, immune responses, cell migration, and stem cell differentiation. And in these functions, they are vital for cellular signal transduction. And because they're so important, if someone has a lipid disorder or their lipids aren't working properly, severe diseases can result, like diabetes or cancer. And lipids have common chemical features such as head groups that are used to distinguish or classify them. And they also have details such as the number of double bonds they have, fatty acid chain length and positioning, ester linkages, and just a whole variety of factors which make them different from other lipids and can be used to put them into groups. And as a result of this diversity of structure, there are thousands of different types of lipids in our cells. Okay, over the last few years, there has been an increasing amount of evidence that suggests that it's individual lipids and not whole groups or classes of lipids that are responsible for cellular signaling. For example, when cellular processes such as cytokinesis occur, a significant increase in phosphatidic acid ether lipid has been measured. So diacylglycerols, or DAG for short, are a type of lipid made when cells need a second messenger at the plasma membrane. Or insulin and ion channel signal regulation, or the release of neurotransmitters. So these lipids also have very important functions. And diacylglycerols can use other molecules like effector proteins to help them perform their functions. For example, they can recruit protein kinase C isoforms, which are responsible for phosphorylation to help control signal transduction. And in recruiting protein kinase C, for example, they can binds to domains like C1 on protein kinase C and use them in their processes. The mechanisms for these recruitments of protein kinases, though, aren't quite known. And that's why this study is being conducted as it is, because individual effectors, like being recruited by individual diacylglycerols, has not been studied a whole lot. So that's what this study is trying to dive into. So the hypothesis of the researchers was that the diversity of lipids is basically the basis for the function of lipids. So similar to how we've heard that protein structure determines protein function, the same could actually be said for lipids when looking at some of the processes that they do. And more specifically, the researchers were investigating the structures of diacylglycerols and how they affected their ability to recruit specific proteins and molecules to assist in cellular processes. For example, their ability to recruit protein kinases was one of the main things they were trying to study here and measure and see how different diacylglycerols would recruit different protein kinases based on structures. So their hypothesis was that diacylglycerol mediated recruitment pattern for a variety of proteins and lipid protein affinities was determined by their structural differences. So the methods the researchers used in this experiment, first off, was caged DAG testing, or diacylglycerol testing. So basis for this experiment was the creation of caged lipid probes. And what this means is that there is a group attached to the lipid probes which inactivates them. But when they are exposed to light, such as ultraviolet light, the groups will be cleaved off and the lipid can function. So this is like an inhibitor almost. And this allowed the researchers to manipulate diacylglycerols in the plasma membrane in a very precise way. They also used mathematical modeling, and what this let the researchers do was combine the information from the uncaging experiments to determine the binding constants for the diacylglycerols to proteins, which allowed for calculation of the parameters for diacylglycerol movement through membranes. And the last major method that they used was live cell quantification. And this was used to image differences in diacylglycerol movement and kinetics, and as well as the differences in protein affinities between them. So images like the one here could be produced through live cell quantification. And this specific one shows the process of diacylglycerol uncaging through the fluorescence. And more on that in the next slide. Okay, so for the results that were obtained, the first experiment was able to find that by using ultraviolet light on caged diacylglycerols, that would result in the diacylglycerols being uncaged and an increase in the diacylglycerol density in the plasma membrane. What the researchers did is they prepared 4 diacylglycerols of different structure, including CG-DOG, and after inserting them into the plasma membrane of living cells, they were exposed to ultraviolet light. In figure F, whole field of view imaging shows that the membrane was bright with dye before encaging the diacylglycerols. And the darkening of the membrane signified that the ultraviolet light was successful in freeing the diacylglycerols because the alcohol group was cleaved off of them, which resulted in the dye being absorbed. And figure G was used to confirm this as the absorbance of the dye in the plasma membrane decreased as the uncaging of CD-GOG progressed, as you can see by the blue line in the plasma membrane. The absorbance was decreasing, so that means the diacylglycerols are being uncaged. And then for this figure, to further investigate how structural differences affected the diacylglycerols, the researchers investigated the effects of uncaging specific diacylglycerols. So SAG, SOG, DOG, and 1,3-DOG were tested in this way because they were all differently structured. And as you can see, the graphs in the figure show that their respective abilities to recruit protein kinases, recruit protein kinases, SAG was the most effective except for alpha and gamma, as none of the diacylglycerols were able to recruit those very well. SOG was also effective in recruiting alpha and epsilon protein kinases. SAG and SOG were also the only long chain diacylglycerols tested here, so them having long chains might have an effect on their ability to recruit protein kinases. It was also found that calcium ions levels were elevated when uncaging the long chain diacylglycerols. So this is also a topic to be looked at. And to further investigate how structural differences affected the diacylglycerols and to find out movement behaviors, the researchers also measured how the diacylglycerols of SAG, SOG, and DOG, as seen in the figure, bound to molecules known as C1, EGFP, and ES. And as the diacylglycerols were uncaged, or as they were liberated, as seen in this figure, the amount of free C1, EGFP, NES decreased, signaling that the diacylglycerols were binding to them, which was also confirming that the ultraviolet light was successful in uncaging. And SAG, if you look at the separate graphs, binds to more of these molecules than that of SOG or DOG, because those seem to level out as more of them are uncaged, but SAG keeps binding to the C1, which is why the C1 concentration continues to go down as the SAG is uncaged. And from the rates of binding to these molecules, researchers were able to calculate the transbilayer movement constants of each of these diacylglycerols and determine the efficiencies of their binding. So SAG was the most efficient because it was able to bind to the most C1. And it also had the most movement because of this. So the significance of this experiment is that the techniques used here were different from many other similar experiments in that they made and injected diacylglycerols before manipulating them, instead of signaling receptors to make diacylglycerols in a cell and then going from there. Because with the method they used here, when they injected diacylglycerols they made themselves, they knew there was a starting point for the amount of diacylglycerols in a given area and the types of diacylglycerols that were available for signaling reactions. And the researchers also found that activating uncaging the diacylglycerols of SOG, SAG, DOG, and 1,3-DOG all had different effects on what protein kinases were recruited for signaling and how many of those protein kinases were recruited. So this implies a high level of specificity between the lipids. And this specificity was because of the different structures in those tested diacylglycerols. For example, diacylglycerols with longer fatty acid chains and a greater number of unsaturated double bonds were seen to be more competent in recruiting protein kinases of many kinds, particularly alpha, beta, and sigma protein kinases. Diacylglycerols with these characteristics also exhibited more recruitment of calcium ions and greater amounts of transbilayer movement upon mathematical calculation, especially in the diacylglycerol of SOG. SOG seems to be the best one for that. So in conclusion, the hypothesis presented in the beginning was supported. It was shown that differently structured diacylglycerols produce different results when uncaging them. In measuring how they recruited protein kinases, calcium, and how they moved through the membrane, they found that the levels of those three factors were different among differently structured diacylglycerols. So lipid diversity was seen to be connected with lipid function. Future research could test a greater range of diacylglycerols over a larger range of factors besides just the recruitment of protein kinases or movement speeds. And they could see how those interactions occurred as well using this experimental model. All right, that is it. Thank you for listening to my presentation.